The next unit in food chemistry is about Browning reactions. Browning is a common color change seen in foods during pre-preparation, processing or storage, which can occur in varying degrees in some foods. These are very complex reactions which occur in food, leading to formation of brown color pigments or brown color, and occur in varying degrees in many foods when they are exposed to different conditions during processing, handling or storage. For the purpose of learning Brownian reactions in foods, we can categorize those Brownian reactions into two types: enzymatic browning and non-enzymatic browning. Non-enzymatic browning includes caramelization, Maillard reactions, lipid oxidation, ascorbic acid oxidation, etc. and enzymatic browning. As a first part, we will discuss about enzymatic browning. The intended learning outcomes of this section is that after learning this section, students will be able to understand enzymatic browning, causes of enzymatic browning, advantages and disadvantages of enzymatic browning, and prevention of enzymatic browning. Enzymatic browning is a chemical process that occurs in plant-based items and shellfish by the enzyme polyphenol oxidase, which results in brown pigments formation. The discoloration that results when monophonic compounds of plants or shellfish in the presence of atmospheric oxygen and polyphenol oxidase are hydroxylated to ortho-diphenols, and the latter are oxidized to orthoquinones. It can be observed in fruits such as pears, apples, apricots, banana, guava, etc. The browning is very obvious in vegetables such as potato, brinjal, lettuce, mushrooms, etc. and in some cereals such as wheat flour and rice. And also we observe the formation of brown color in seafoods, shrimps, crabs and spiny lobsters, etc. Enzymatic browning is detrimental to quality in fruits, fruit juices, vegetable juices, and some shellfish, especially in their post-harvest storage. It can be responsible for up to 50% of post-harvest losses during fruit and vegetable processing. Enzymatic browning, on the other hand, is an essential step for color and taste development of some foods such as coffee, tea and chocolates. The enzyme involved in enzymatic browning is copper containing enzyme that catalyze the oxidation of phenol compounds contained in plant tissues or in shellfish. Enzyme polyphenol oxidase act as a catalyst and speed up the process at a rapid temperature when the pH is between 5 to 7. The optimum activity we observe near the pH 6 and 7. pH near 7, the optimum activity is observed. This enzyme is fairly resistant to heat, that is polyphenol oxidase. The polyphenol oxidase, the enzyme responsible for enzymatic browning, is fairly resistant to heat and its activity is optimum near neutral conditions. This enzyme acts on monophenol or orthodiphenol, not on meta-diphenol. Reactions in enzymatic browning, we can categorize this polyphenol oxidase reaction or polyphenol oxidase catalyzed reactions into two basic reactions, which are hydroxylation followed by oxidation. The phenol is catalyzed therefore two types of reactions. First, hydroxylation followed by the oxidation reaction. Usually in plant cells, this enzyme localized in plastids, while substrate, those are phenolic compounds which are in vacuole. In natural conditions, these two components, substrate and enzyme, will not meet, but when we perform processing or cutting, abrasion or damages to cells will lead to the contact of enzyme into its substrate, making it allowed to form brown color pigments. So as we discuss now, we'll see what are two type of reactions which catalyzed by polyphenol oxidase. The first one is hydroxylation. So we can call it as cressolase activity followed by the catecholase activity, which is an oxidation reaction. In cressolase or hydroxylation reaction, during the hydroxylation reaction, phenol hydroxylase of cressolase activity results in orthohydroxylation of phenol. This reaction results orthohydroxylation of phenol. So the reaction is hydroxylation. The reaction is a hydroxylation. Then we have second reaction, that is oxidation reaction. That is oxidation reaction. Catecholase activity. So this acts as a dehydrogenase enzyme. These two enzymes, catecholase and cressolase, this is kind of enzyme which is leading to hydroxylation, and then catecholase enzyme which is dehydrogenase enzyme. So it removes hydrogen from substances. This reaction therefore results in the oxidation of diphenol to an orthoquinone. So this result, oxidation reaction. So phenolase catalyzes two types of reactions. First one is hydroxylation followed by the oxidation reaction. Here you see an example of polyphenol oxidase reaction. So here you see the formation of tyrosine, formation of melanin from the substrate tyrosine. So as you see, as you remember, the polyphenol oxidase catalyzed two kinds of reactions, that is hydroxylation and then the oxidation. These both reactions utilize molecular oxygen as co-substrate. You see both reactions. both reactions utilize molecular oxygen as co-substrate. So it utilize molecular oxygen as co-substrate. So this is phenolic compound, the tyrosine and amino acid. It is phenolic compound. The first reaction is the hydroxylation. So where oxygen is a co-substrate and the enzyme converted it into dihydroxyphenylalanine. Dihydroxyphenylalanine or DOPA. Then this DOPA is followed by the second kind of enzymatic reaction, that is oxidation. DOPA oxidized to dopa-quinone. That is oxidized to dopa-quinone. So this is the enzymatic reaction part. In this complete reaction, so we see in this conversion, the formation of brown color pigments, the first part is catalyzed by oxygen enzymes. The second part is non-enzymatic oxidation reaction. So in conversion of tyrosine to melanin brown color pigments, the first is the polyphenol oxidase activity. The first reaction is hydroxylation, which results the formation of dihydroxyphenylalanine or DOPA. DOPA is then subsequently oxidized to produce dopa-quinone. This DOPA, this halochrome, which is red in color, this halochrome may undergo decarboxylation. This can undergo decarboxylation reaction and get changed into 5,6-dihydroxyindole. 5,6-dihydroxyindole and 5,6-dihydroxyindole 2-carboxylic acid. 2-carboxylic acid. So this DOPA, which is halochrome, this can further undergo reactions. This halochrome, red in color, it can further undergo reactions and then converted into 5,6-dihydroxyindole or 5,6-dihydroxyindole 2-carboxylic acid. And ultimately both this 5,6-dihydroxyindole and 5,6-dihydroxyindole 2-carboxylic acid, both are converted into various unsaturated carbonyl compounds, which are collectively known as melanin. So these are known as melanin compounds. So this is an example for enzymatic browning, the formation of melanin compound by tyrosine. So this is the phenolic compound. So the first part of this conversion is enzymatically catalyzed. The second part is non-enzymatic oxidation reaction. So in enzymatic conversion, tyrosine first undergo hydroxylation yielding DOPA, and DOPA can oxidize. It will undergo oxidation reactions with the presence of oxygen as a co-substrate. You see in both reactions, oxygen is a co-substrate, and that dopa-quinine is produced. Ultimately, this leuco compound also converted to halochrome. This dopa converted to halochrome. This halochrome, dopa converted to halochrome, and then dopa-quinone also converted to halochrome. This halochrome can undergo various reactions yielding two kinds of intermediate products, 5,6-dihydroxyindole and 5,6-dihydroxyindole 2-carboxylic acids. Both are converted finally into various unsaturated carbonyl compounds which are commonly called as melanin. So you see that latter portion of reaction sequence involves non-enzymatic oxidation. We'll see what are the advantages and disadvantages of Brownian reactions.